Welcome back, folks. This is a video lecture on energy carriers. The agenda that we're going to utilize today is pretty straightforward. We're going to do a quick recap on our understanding of energy. Then we'll move on to talk about how energy is handled inside cells. In particular, we're going to focus on redox reactions. A quick recap on that particular phenomenon. And then we'll talk about 5 types of energy carriers that are very important to living systems. Of those, we'll focus mainly on ATP, since it's the universal energy carrier of most cells. And then we'll talk about 2 of its lesser known cousins, NADH and NADPH. And then we'll wrap everything up into a understanding of how energy carriers, and metabolic reactions are related to forming both catabolic and anabolic reactions within living cells. Let's get started with the first of these. As we know, energy cannot be created or destroyed according to the laws of physics. Therefore, everything that we do in biology has to abide by those principles. But energy can be interconverted from one form to another. And as you know, every time energy is manipulated, there's a small tax to pay. And that tax is what we call heat energy, or more precisely, entropy. The first law of thermodynamics is simply a statement reinforcing what I said earlier, that energy is a constant. It cannot be created or destroyed, but it can be converted to other forms. And every time you do, there's some type of penalty. And that penalty goes with the second law of thermodynamics, which says that entropy is generated whenever energy is manipulated. And entropy can be looked on as disorder, chaos. As you know by now, cells are highly ordered in their chemical infrastructure. So do cells violate the laws of physics? Well, they can't. Therefore, every time a cell takes material and orders it inside, therefore there must be a release of entropy into the surroundings. And that entropy then adds to the chaos of the universe. Pretty straightforward. So if those are the boundaries in which living cells operate, then how do they manipulate energy? Because they can't destroy energy or make energy. So these interconversions always result in the generation of useless energy, as physicists call heat. You and I know heat as something slightly different. But let's just stick with entropy, because that's a real definition of what is actually happening, right? So every time you move your arm, every time you use your eyes, every time you think, you're taking energy within your cells and converting it to a different form. And that generates entropy. Living things need a constant supply of energy. If a cell goes without energy even for a few milliseconds under normal circumstances, the disorder inside the cell will rapidly increase and the cell will die. For us humans and heterotrophs in general, we get our energy from pre-made molecules, food molecules. So by consuming those molecules, we can break them down, releasing their energy and utilizing their energy to maintain or build new structure. So how does food supply us with energy? Well, food molecules contain electrons, and those electrons come in various orbitals. So the low orbital electrons contain less energy than the high orbital electrons. And by moving these electrons between molecules, their energy can also be transferred. So electrons are the primary way in which energy in molecules is handled by biological entities. In addition to just electrons, protons also possess energy. And wherever we have a release and high concentration of protons, we will have associated energy. So by moving protons from one place to another, we can transfer energy. And finally, the alternative is to form high energy bonds between molecules. And these bonds, they come in different strengths, and some bonds happen to have more energy stored inside them than other bonds. By dismantling the molecule or breaking these bonds, that energy, or at least a significant proportion of it, can be then utilized by the cell, and that drives the living processes. And if you remember all three things, we'll be handling these using the different types of energy carriers which cells possess. But before we can do that, let's have a look at redox reactions. Very briefly, these reactions allow us to follow where the energy is. As we said, if we follow the electrons, we can follow the energy. So suppose we have something that carries a lot of energy. Everybody should know that on your gas cooker, when you light the flame, you're burning a hydrocarbon. And that hydrocarbon happens to be methane. So methane consists of one carbon bonded to 4 hydrogens. And as you know, that can be explosive under certain conditions. So there must be energy stored within that molecule. Where is the energy? Well, as we just said, the carbon is bonded to 4 hydrogens. And whenever you mix this with oxygen in the atmosphere and a spark to get the process going, you get an explosive ignition of the gas methane. And the bonds between carbon and hydrogen are broken, and the rearrangement of atoms takes place during the reaction, such that the end product of the reaction will be carbon dioxide and water. So what's going on? Well, we can relay to you the manipulations that are taking place between the electrons of these various atoms. So let's break that down very quickly. Let's get rid of some of the writing on this slide so we can use the space to relay ourselves with the activities going on inside redox reactions. So first thing to remember is reduction is a term related to electrons. So reduction is gain of electrons. So whenever an entity has more control over its electrons, or it gains new electrons, that's the key to understanding redox reactions. And the opposite of that is, where did those electrons come from? So the entity or the atom or the molecule that loses control over its electrons in this single manipulation is said to be oxidation. So oxidation is loss of electrons. And you can't have one without the other, because electrons cannot just come into existence. They have to be pre-existing and then just transferred around. It's like us transferring money between us. If I have the money and I give it to you, you gain the money, I lose the money. So redox reactions go hand in hand. So one entity has to gain electrons at the same time as another entity losing control of electrons, or at least control over those electrons. If the electrons are closer to me than to you, I have more control over those electrons. If after the reaction, those electrons are closer to you, then you have control over those electrons. And that's the way to look at it. So let's apply that to our example of burning gas. So we have carbon with hydrogens, and that's methane. And in the case of carbon and hydrogen, if you look in a periodic table of electronegativity, you will find that carbon pulls ever so slightly more on the electrons in the bond between methane. So carbon, hydrogen, there's 2 electrons here, and there's another hydrogen there with 2 electrons, one more hydrogen there, and a final hydrogen there. And those 8 electrons are pulled closer to the core, the nucleus of carbon, than they are hydrogen. Due to its electronegativity, the carbon atom not only has its 4 electrons that it came with, but it gains control over the 4 electrons that belong to those 4 hydrogens. So in reality, it's gained control over 4 more electrons. So it has an overall charge of -4. At the same time, each hydrogen came with its own electron, but that electron has been sequestered by carbon. Therefore, each hydrogen has lost control over its electron. So from a redox potential, it has a plus sign, each hydrogen. So those 2 should cancel out. Just like energy, you can't make or destroy electrical charge in this manner. And then when you combine that with oxygen in the atmosphere, oxygen occurs as a molecule. And those molecules have control over electrons. And when you have 2 elements the same as in molecular oxygen, each oxygen pulls on the electrons equally because they're both identical. And the electrons will be found at the center of the covalent bonds. In this case, neither oxygen has gained or lost control over any of the electrons that it had. So we put a 0 here. So its redox potential is 0. And then during the reaction, we form carbon dioxide and water. So now let's look at the electronegativity of carbon. So carbon dioxide is a molecule that looks like this. And oxygen has a much more electronegativity than carbon does. So the electrons will be found now closer to the oxygens than the carbon. So in this new relationship, the carbon has lost control over the electrons which it had inside methane. All the electrons are lost, the orbital electrons. So this time, not only has it lost control over the 4 electrons that belong to the hydrogens, but now it's also lost control over the 4 electrons that are present in its outer orbital. So in that sense, we now have a reversal of charge. So each carbon is now 4 redox potential positive. Each oxygen has gained 2 extra electrons that belong to carbon. So that's going to be 2+. And because there's 2 atoms of oxygen, that adds up to 4. So again, the molecule itself has a 0 net charge, but each part of that molecule has been manipulating the electrons. And of course, in water, water looks like this, with 1 hydrogen bond. Sorry, 1 covalent bond to each hydrogen. There are no hydrogen bonds in water, as somebody mentioned to me recently. They aren't. Hydrogen bonds are between molecules, not within the molecule. So these are covalent bonds. And in the covalent bonds, the electrons are going to again go towards the electronegative atom, and the oxygen gains control over those electrons. Therefore, each oxygen will be 2-, and each hydrogen is a plus. And again, that molecule of water balances out. So with respect to carbon, what's happened? Has the carbon been oxidized or has it been reduced? And the answer is simple. Carbon had a -4 redox potential there. And then over here, carbon now has a +4. So there's been a change. And because carbon has lost control over its electrons, loss is oxidation. The carbon has become oxidized, and the oxygen has been reduced. With respect to hydrogen, nothing changed. You'll notice that the molecule here has hydroxides, and the molecule down here only has oxygens. So pertaining to the carbons here and here, what's happened to this molecule? Which is the oxidized form and which is the reduced form, right? So oil rig will help you, and the addition of oxygen should give you an idea of what's happening. Let's move on rapidly to activated energy carriers. So what are these things? These are basically transporters. These are molecules that have been sequestered by chemistry and biological systems because of their properties in being able to carry easily and unload easily these energy carrying electrons and protons. Okay, so the transporters are designed just like minicabs to pick up passengers at a place where energy is being liberated, and then capture the energy, take it to a destination where the energy is required. They can hang on to and store the energy temporarily as well. Not just electrons or protons can be carried by these specialized activated carriers. They can also carry molecules, high energy molecules. And we have all three types present within our cells in many varieties. In fact, if you look at the most common ones, which are listed here, ATP is present in millions and millions of individual molecules. Therefore, every second, lots and lots of ATP is being rapidly turned over to supply and capture energy within the cell. So it's a hive of activity within our cells when it comes to activated energy carriers. So ATP, what does it carry? Well, it carries a chemical group. And we'll look at that chemical group on the subsequent slide. In contrast, both NAD and NADP, they both carry electrons and they can carry protons at the same time. So 2 and 3 are equivalent to a taxi cab that can carry passengers in the back seat and in the passenger seat on the front. Maybe the proton gets in the front seat and the 2 electrons get on the back seat. But whatever analogy that you use, make sure you understand how that analogy then relates to electron carriers. Right? There's my attempt to draw a taxi cab, not the best. I won't get in that taxi cab, but you get the idea. So the proton will jump in the front seat and the 2 electrons will jump in the back seat. And then this taxi cab will go from a loading site to an unloading site where it will unload the passengers. And just like any other taxi cab, it can recycle back and pick up more passengers, either from the same location or any other location within the cell. So the analogy to a taxi cab really does work well in this particular example. These two are direct cousins of each other, and we'll talk about them very shortly. Then there's a more distantly related cousin called FADH2, and that is also an electron carrier, and it can carry 2 electrons plus 2 protons. So slightly different, but the analogy to a different type of energy carrier or cab company will work there too. And it does exactly the same thing as its 3 brethren above, ATP, NAD, and NADPH. Now you notice that I haven't mentioned the H when I was talking about 2 or 3, because when the H is present, that means the taxi cabs are occupied. When you remove the H and you get NAD+, then that means the taxi cab is unoccupied, right? So you can go from NAD plus, when you have a vacant taxi, to a full taxi, NADH. And then, of course, the taxi, once it unloads its cargo, can come back and become empty again for another round of passengers. So all three of those molecules, 2, 3, and 4, they're able to do this. And then finally, we have many more, of which we've just chosen one, acetyl coenzyme A. And this carries a molecular group that's added onto the molecule when it's carrying energy. And that molecular group can be disengaged when it gets to a destination, and that energy can be then delivered to another molecule. And we won't go into details on that. So let's look at this universal use of ATP. We believe that ATP, which is a modified nucleotide, and I'll draw that in a second, it came into existence as an energy carrier early on in the history of life. Therefore, it's not surprising that the vast majority of cells, if not all cells, use ATP as their primary source of handling energy and moving it around inside their protoplasm. ATP is the charged version of the molecule. So ATP stands for adenosine triphosphate. The adenosine is a reference to this particular base. So ATP is a tiny little molecule that is made of 3 parts. It's made of a base, and in this case, the base must be an A, otherwise its name would change, adenosine. And it's bonded through covalent bonds, right here you can see it, to a sugar. And the sugar is a carbohydrate, of course. And then we have a number of phosphates. And if you have 3 phosphates, as in this example here, then it's called a triphosphate. And that becomes ATP, adenosine triphosphate. If you break one of these bonds, this one here, and as you can see down here, then the inorganic phosphate will float away, leaving behind just 2 of the 3 phosphates. So that becomes a diphosphate. Right? So there's going to be ADP. And of course, this bond here can also be broken. And once you break that bond, then you have just 1 phosphate left attached to the sugar, and that's going to be your monophosphate, or AMP. Okay? Not all bonds are equivalent in chemistry. Some bonds release more energy when they're broken, covalent bonds, than others. And in this particular case, for some reason, because of the properties of the phosphate and the oxygens and being so negative, these bonds are special bonds. They're very high energy bonds. When you break these bonds, you release a lot of energy. Likewise, when you make these molecules, you have to have a lot of energy supplied into this. So your food supplies the energy when it's been deconstructed, when it's been broken down by your cellular machinery. And the energy in the food is used to power the production of ATP in many steps. So when you produce ATP from ADP, you're going in this direction, and that's when you're storing energy within these activated carriers. And when you go in the opposite direction, you're then releasing some of that energy to perform some type of activity within the cell. And of course, a small amount of that energy will be released as entropy, as useless energy. So every time you do a manipulation, even in this direction, you will lose some of your energy as entropy. Thus, a constant supply of energy is needed by the cell. The name of those bonds, these bonds here, are phosphoanhydride bonds. Phosphoanhydride. Very powerful, carry a lot of energy, and maybe they are instrumental to the success of modern day organisms. So phosphoanhydride bonds. Since we're talking about ATP, ATP is such a versatile molecule that it can be utilized within the cell by two alternative pathways. In the first, the breaking of that phosphoanhydride bond, either the first one or the second one, it doesn't matter. Each one releases the same amount of energy. The breaking of either one of those bonds can be used to directly drive a reaction that requires energy. And a reaction where energy needs to be supplied in order to make that reaction work. So that's what we say, by just breaking the bond in the vicinity of the other molecule, that molecule can then capture some of this energy and perform its manipulation. The alternative way that ATP can supply energy is by using the terminal phosphate and attaching that to another molecule. So suppose you have another molecule, let's call it M, and this molecule is in the vicinity of ATP, and then you break the first phosphoanhydride bond. The phosphate can then be transferred to this molecule here. And this energy bond, this bond here that's formed between the molecule and the phosphate, is a high energy bond, because most of the energy from ATP is taken with the inorganic phosphate to this other molecule. And now this molecule here carries more energy than it did prior to this exchange. And then later on, this energy in this bond can be used to drive a subsequent reaction or some other manipulation in constructing something within the cell. So ATP can be utilized in two fantastic ways. Let's move on quickly to look at NADP and NADPH. Well, that's not right. So this should say H. So students do this all the time, and I missed to doing that myself. So I mislabeled that, so forgive me. But I think in the agenda I got it right, and here I got it wrong for some reason. But let's move on. So these two are alternative ways of carrying energy within the cell. NAD is able to pick up energy in the cytosol from the initial breakdown of glucose. So as you're dismantling the glucose sugar molecule, some of the energy is released. Some of the energy is released in such large packets that you can use it to make ATP from ADP. But in most cases, the energy is released in less than total units. So there's not enough packets of energy to make ATP, but these packets are a bit smaller. And instead of wasting this energy as heat, NADH is formed from NAD+. Right? So NAD, this is a typical cell. And then we have the nucleus, and we have mitochondria. Mitochondria are very important. So that's the nucleus, and that's the mitochondria. So when sugar enters the cell, glycolysis, let's get the spelling right, glycolysis takes place in the cytosol. And here we have lots and lots and lots of NAD+, waiting around for passengers. And electrons are liberated from the various steps of glycolysis, and NAD+ picks up those electrons as well as protons, and it becomes NADH. And then it goes and makes its way, sorry, that's the wrong one. It doesn't go into the nucleus. Forgive me. It doesn't go into the nucleus. The NAD takes a journey and it goes into the mitochondria, okay, where it delivers those electrons for oxidative phosphorylation to make ATP in another way. So understanding the total infrastructure of a cell does assist you in understanding the individual functions of these electron carriers. The diagrams that you see on the right-hand side of the slide, this one here is NAD+. NAD+. This one at the bottom is its cousin, NADP+. What's the difference? The difference isn't that great. If you look at the bottom of this molecule Let me change the color so we can see that. If you look at the bottom of this molecule in this vicinity here, you will see that we have 2 hydroxides on the bottom sugar. And if you look at the same location here, you will see that one of the hydroxides has been replaced by a phosphate group. And that's where the P comes from. And that's all it is. And we'll explain why there's 2 versions of this same, almost identical molecule. But let's focus at the top of the molecule, the top end of the molecule here. Okay? This is where the passengers are picked up and carried within those nitrogen rings. So the insert over here that we have, this insert can be applicable to both NAD plus and NADP plus. This part represents an upside down version of this. So these 2 are the same, except that one is flipped upside down. So the rest of the molecule is here. That would be the rest of the molecule that you see here. Okay, it's a bit messy, but that's okay. Let's change the color so we can see what's going on. So what we're going to see is what happens to this ring. And this is the same ring. So here's the general equation, NAD plus, which is this form here. Then in the appropriate location within the cell, when electrons are liberated by a chemical reaction within the vicinity, electrons, 2 of them, are picked up, as well as a proton. And you can see, if you look very carefully, you'll see there's a plus sign here originally next to the nitrogen, and that plus sign has disappeared because of the capture of one of those electrons. It neutralizes the positive charge. And the second electron combines with the proton, and then it forms a hydrogen. And there's your hydrogen. So between this and this, that's where the passengers are now on board this molecule. And exactly the same thing happens with NADP plus and NAD plus. Once this molecule or these molecules get to their destination, the passengers can be unloaded, and you can see the arrow telling you that the electron carrier can then go back to its normal accepting. Which of these 2 forms is oxidized and which of the 2 is reduced? So remember, oxidized is when it's losing electrons, and reduced is when it's gaining electrons, right? So this side has more electrons than this side. So this side, the molecule has more electrons, so it's being reduced. Reduction is gain. So this is the reduced form of NAD, and this is the oxidized form. So now you're applying your understanding of redox reactions to these. Before we leave this slide, let's look at these 2 statements. It says that NAD plus is normally used within the cell for catabolic reactions. Catabolic reactions are those reactions that break down, for instance, food molecules into carbon dioxide and water, and is preferred in those circumstances. Whereas its cousin, NADPH, is used for anabolic reactions, reactions where maybe the food molecules are built up into something more substantial, like proteins and DNA. So by having 2 separate molecules within the cell, and each one can be either in the oxidized state or the reduced state, it gives you 2 independent systems that don't couple the 2 types of reactions together anymore. So you can do your anabolic reactions independently of your catabolic reactions to a large degree. So it gives the cell more variety in order to perform these type of manipulations without having to keep an eye on how much of that do I have and can I make an equivalent amount. So there's a lot of independence. And that's what these activated energy carriers do. The more interdependence there is, not interdependence. So that's very important. So let's finish off quickly by looking at a totality of everything we've learned so far in terms of energy and what drives cells, right? So here's a cell, and let's make it a eukaryotic cell. Prokaryotic cells will be under the same constraints. So we have reactions when we have food coming into the cell, food enters, and 2 things can happen to that food. the food can be broken down, right? So breakdown. And the ultimate in the mitochondria will be carbon dioxide and water. And these are highly, highly oxidized forms of molecules. So they contain very little energy. So carbon dioxide is basically one of the lowest forms of molecule that living cells have to deal with. Of course, water is its counterpart. So when you take food like glucose and you break it down through various steps and various pathways into carbon dioxide and water, you have a lot of energy being released. And some of that energy is used to make ATP from ADP, right? So the energy is stored directly in the ATP, and that can be used by other parts of the cell. And some of these release protons and electrons, and those can be picked up by alternative types of energy carriers. And then they can deliver them to a destination. So this pathway here, the breaking down pathway, is called catabolic. Overall, it releases energy. By releasing energy, we call those exothermic reactions. Not every step is exothermic, some require energy, but in overall, they release energy. So this side of the cell is releasing energy. Now, everything is intermixed in a real cell, but in our diagram, we can say that. And then the food molecules, some of them, can be taken under certain conditions and used to build up more complex chemistry. Even more complex than the amino acids, say, that are coming in with the food, right? So cells receive in your body amino acids in the blood that are then delivered to the cytosol. And in the cytosol, they can be then built up into proteins. So proteins are more complex. They contain peptide bonds, and then they fold into whatever configuration they need to. So that's a building up type of reaction. So building up reactions are called anabolic. And in general, they are endothermic reactions. That means you must supply energy in order to force the food molecules to be rearranged and then combined into more complex structures, right? The other thing is we have nucleic acids. They can come in through the diet, or they can be manufactured inside the cytosol. They too can be built up into DNA and RNA. And those are also anabolic reactions. And you can take simple sugars and build them up into more complex sugars, right? Like glycogen. And that would require glycosidic bonds to then build that larger structure. And the larger structure will be then held together by these glycosidic linkages, which are covalent bonds, and therefore they have energy stored in them. In the future, if you break those bonds, you'll then dismantle the molecule and then release that energy, well, most of that energy, into the cell, okay? And the same thing with lipids. You can take simple triglycerides and then make them even longer by making their fatty acids longer, or you can rearrange those molecules to make more complicated molecules like phospholipids. And that also would be a form of anabolic interaction. So you get an overview of what's going on inside the cell, right? So just to finish off, exothermic reactions, these are reactions that release energy into the environment. Now, do they have a positive or negative delta G? So if you add something to the environment, are you having a positive or negative delta G? And the answer is, if you release something, you're going to be negative. So you're contributing energy away from the molecule, the minus, into the environment. And that energy can then be either wasted as heat or used for capture by activated energy carriers, or to directly couple to a biosynthetic reaction, right? A endothermic reaction. And then over here, we have delta Gs that are positive. That means you have to supply energy into the system, and then it will build up. So we now have concluded our understanding of anabolic and catabolic systems. And hopefully you're in a better place to know what happens. Thank you so much.